Okay, what is the first week of a science class if not for the scientific method? This is how you know you're a biology major, because most of your classes are going to talk about this. So basically, we're going to identify the problem. We're going to form a hypothesis for the cause. We're going to test the hypothesis. We're going to collect data, interpret the results, and report the results. What this looks like in plant science right now. My job, particularly, is to clean up the Mississippi River. So that's a problem. The Mississippi River is gross. I had a hypothesis. I think stream banks and ditch banks and the erosion that we're seeing there from high rainfall events is contributing to this fertilizer and other things in the Mississippi River that are causing a hypoxic zone in the Gulf of Mexico. So our hypothesis was stream bank erosion is causing some of the issues. So in order to test that hypothesis, we went to 25 stream banks in Illinois and Missouri, and we spent 3 years looking and recording the soil loss and where that soil was moving through streams into the Mississippi River. We collected that data, we interpreted the results, and now we are sending those results to different peer-reviewed journals. Let's look at soil compaction. So a controlled experiment. Essentially, you're going to want to randomize this if you're going to do a controlled experiment. This is one of two ways. My stream banks, we couldn't do that. But if I wanted to look at soil compaction, if I wanted to look at the effects of fertilizer, if I wanted to look at yelling at plants, if I wanted to look at anything like this, I would do a controlled experiment and I would randomize my pots. This is pretty easy to do in the greenhouse. You're going to set up a situation where only the factor controlling the outcome is the one of interest. So in this case, we're looking at plants in compacted soil. Compacted means the soil doesn't have enough roots to spread it out, and so it's hard for the roots to get down. So you've shoved that soil into a pot and kind of pushed it down there, and you planted a seed on top. And in the non-compacted soil, you would loosen it up and maybe put some perlite in there and make it to where the roots could move around a lot. So in order to test that theory, you would plant a lot of pots, and some of them would be compacted and some of them wouldn't. Why have them separated out like this? Why not just put compacted on one side and non-compacted on the other side? Basically, that's because if this is your greenhouse and the light is coming in here, and if you put the compacted right here and the non-compacted over here, how do you know that the effects from the light, or maybe there's an air conditioner valve here, or maybe, you know, the janitor comes in and knocks it over every day, or you have no idea. So one of the reasons that you want, especially in plant science, make sure that your pots are randomized so that you can make sure that the variable you're controlling is what is causing the plant to get taller, bigger, greener, and not some other factor like watering or light or, you know, the effects of shading from the plants next to it. That's also something that you have to consider in plant science research. Another way of testing a hypothesis is more descriptive research. So essentially, this is a large-scale hypothesis, such as my cause for erosion, or you're isolating part of the problem. So one of the things we do in plant science a lot is we get these, it's a PVC square. So you would go into a field and you would kind of randomly throw it down like a frisbee, and then you would count the number of plants in this area, or you would take a sample of biomass. Here they might be cutting all this grass and weighing it. And then you would take this 1 meter area and extrapolate that to the entire rest of the field. So you would say, in a 40-acre field, there's this mass of wheat or this mass of weeds. Or maybe very often you would count the number of plants in here and you would say there's 20 plants in this meter. So in a 1-acre field, there's 10,000 plants. So that is one way in which you would take data where you're extrapolating from a smaller area where you're taking the data and you're extrapolating it to a larger area. This is what they've done here. It's called a correlative study. They've conducted on a small area and made correlations to a larger area. This section of the field has a small tributary stream in the example on the slides. And that section has a stream running through it, and it can be examined closely for the effects of the farming practices. And then you can interfere that. You can use a modeling, or you can use some other way to say, okay, this is what we found in this section, and we're going to extrapolate that to the other sections and make some kind of prediction. Okay, back to the controlled experiment. For a controlled experiment, you're going to have two concepts, the independent variable and the dependent variable. The independent variable is the one being tested, the treatment. This is for sure going to be a multiple choice question. You got to know this is coming. I might even make you design an experiment in an essay question. The dependent variable is what changes from the independent variable's treatment. So soil compaction is the independent variable. Everything else is the same. We compacted the soil. What we want to know, the dependent variable, is do plants grow better? So in order to do that, we would take the height of the plant, the weight of the plant, the greenness of the plant. We would take dependent variables so that we could say soil compaction decreases plant growth. Soil compaction, the independent variable, affects crop growth, the dependent variable. To help ensure that only one factor of interest is affected, several techniques are taken into account. One is what I just said about randomization. You really want these plants to be as random in the greenhouse as possible. You want all of them spread out, and you want to make sure that you're really seeing the effect from the treatment and not the effect from just one was closer to the wall. Now, another thing that you will see is what's called blocking, a randomized complete block. So imagine each one of these numbers right here is a bench in the greenhouse. So in the first bench, I have fertilizer. I have a control treatment, 20 tons of fertilizer, 40 tons of fertilizer, 60 tons of fertilizer. Not whole tons, but I've extrapolated down to it was probably like 3 milliliters in a gardening jug. So this is one bench in the greenhouse. Then maybe a few feet over, I do another bench in the greenhouse, same treatments. Then I do potentially another bench, and then I'll do a fourth bench. You see they're also in varying areas. This really helps me tightly control and make sure that the effect that I'm getting, bigger plant growth in this case, is based on the treatment and not just it was at a different space in the greenhouse, right? I have done this on 4 different benches, or in some cases 4 different fields, or 4 different places in a field. And I have subdivided that so that each block gets all the treatments and they're randomly placed within that treatment. That's called a randomized complete block. And in plant science research, you will almost always see that because it takes some of the variability out of it in terms of like where the soil might be different or where there's a low spot. You also might see that with different plant heights because when you grow them in the greenhouse, you're still some are just gonna grow differently. So you want to put like, when you start the treatment, maybe you have a taller one and a shorter one, but they're all in one bench and then another bench so that you're accounting for that variability. Another reason to do a block design. So if you look at this field, I'm gonna move this one over so you can see it a little bit. Hold on. Move this one over here because it's easier to explain. You've got some shaded areas here. But you can see where your blocks are. This is a block. This is a block. This is a block. This is a block. So you're going to know if it's the treatment affecting it and not just this shaded area or this low area or this wet area. That's what the randomized complete block is for. Data collection, many ways of doing statistical analysis. Most is designed to give percent probability that only the independent variable, because we only want to know if fertilizer or compaction are affecting it. We don't want anything else to affect the plant height or the plant growth or how green the plant is. So we usually try and say we are 95% confident that the effect of plant growth is from compaction, is from fertilizer use. Otherwise, we would say the results are inconsistent. We have too much variability in all the treatments, and we can't determine if it's actually from what we've treated. Probability determination starts by getting the averages of the treatment and the control and comparing them. But averages do not tell us a story all the time. Essentially, if you think of a classroom, has this ever happened? So it happens in math, maybe calculus, maybe organic chemistry. The class either fails the exam or they get an A, and there's no in between. Nobody gets a B, C, or D. You either get an A or an F. The average of the exam is a C, but that doesn't reflect anybody's grade. That's why sometimes averages are not helpful if there's a lot of variability in the data. So, for example, assume we had one treatment where the grams were 12 grams, 10, 11, and 13, and then 12, 6, 5, and 14. Well, it looks like there's a difference, but what's going on with the 6 and 5? Maybe there's something else happening that isn't related, because this 12 and 12 are the same and this 14 is larger. So even though the averages are different, it feels like maybe that's not a treatment effect. So percent probability actually considers variability of the numbers and makes up for those averages, so you can see what kind of variability you have in the data. And ideally you want low variability. So you really don't want the 6, 5, 5, 14. That's high variable data. And I would not be sure that my treatment was the effect of that because the data is so variable. But if you go here, you can see that treatment A is 12, 11, 11, 12, and B is 9, 10, 9. Always lower, consistently lower. This is consistently higher. So that's more likely a product of the treatment. More likely, treatment A, if you put fertilizer on it, the reason these are bigger is because they had fertilizer. Treatment B is smaller because they didn't have fertilizer. So that's a little bit about variability. Most reports of scientific inquiry, they go through the peer review process. So any kind of scientific literature is going to go through several peers. Usually if you publish in a reputable journal, it's going to go through between 4 and 8 people before they agree to publish it. And that will usually go to other scientists. Now, in botany, plant biology, agronomy, horticulture, you're going to have trade journals. These are the journals that are actually going out to farmers, going out to horticulturalists, going out to people that work at orchards. These are very often taking that research and putting it into an article in a way that is more informative for the average grower. So the researcher would publish on their results of fertilizer, and then somebody in a trade journal would take that and say, hey, you should be using, this fertilizer at this amount for the best results on your plants. So that is sort of the difference. In agronomy, horticulture, you're going to have trade journals that are giving you extra information. Again, reports on experiments are going to go in scientific journals, and reports in trade journals are going to go out to actual growers of these kinds of plants. Okay, that's it, chapter 1.